Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershteyn. Hello. Tonight our guest is Yuri Deigin, founder of YouthBio Therapeutics and a longtime activist in the longevity field. Yuri comes from a background of pharmaceutical drug development where he led a number of biotech companies. He was one of the earliest believers in the potential of partial reprogramming as an approach to slowing down or even reversing aging, with his first foray into the translation of this approach in 2017 and continuing with it to this day. Yuri, great to have you with us.
Thank you very much.
So tell me, what is partial reprogramming?
All right, straight to the chase, huh? So partial reprogramming is this novel technology for rejuvenation, novel approach to rejuvenation, where we use reprogramming, cellular reprogramming, that is induction of Yamanaka factors, but partially. Like Yamanaka factors are these magical factors that were discovered by Yamanaka and Takahashi in 2006 to be able to reprogram cells back to essentially embryonic state. They call it pluripotency. And so this discovery was a big novel discovery for the field, because until then it was thought that cell fate was irreversible. Basically, cells after your initial egg was fertilized, which you were at conception, just one cell, and then cells differentiate and they become, skin cells or brain cells. It was thought that they cannot go back to this embryonic state. But Yamanaka and Takahashi's discovery changed that. They showed actually no, this is a reversible process, and you can actually take a skin cell and reprogram it back to embryonic state. And also, in the process, other people discovered that during reprogramming, cells are actually rejuvenated, physiologically rejuvenated. And for us, for the aging field, this was kind of the crucial aspect of reprogramming that we're trying now to repurpose to turn into therapies that we can do rejuvenation, but without the full reprogramming part. So that's the partial part of reprogramming. Basically, we're activating these Yamanaka genes, but only for a short duration, so the cell does not actually irreversibly go back to this pluripotency, but remains the same cell type that it starts with, but it's rejuvenated in the process. So in a nutshell, this is what partial reprogramming is in the context of rejuvenation.
Why wouldn't you want to fully rejuvenate the cells?
Right, well, you definitely want to rejuvenate them as much as possible, but you definitely do not want the cell to fully reprogram, because then it loses its function, it loses its cell identity, and the organs, basically, which consist of terminally differentiated cells, will no longer function. Because, if the cell stops doing its job and kind of turns back into this pluripotent cell, the pluripotent cell cannot basically do the function of a skin cell or a brain cell. Its function is to just proliferate. And so that's one aspect why you don't want it. The other aspect is that if the cell actually goes back to this pluripotent state, it can then actually start dividing and potentially cause these tumors. They're called teratomas, which are also not a good thing in the context of an animal. So we need to prevent that. We need to prevent cells from actually reprogramming fully. And we have this sort of like a Goldilocks zone where we already get some rejuvenation during the reprogramming process, but we still don't cross this kind of Rubicon of full reprogramming, where a cell, if it crosses it, basically it's going to turn pluripotent. So we operate in this kind of therapeutic window of rejuvenation, but not yet loss of cell identity. And then when we stop the induction of Yamanaka genes, the cell then it remains rejuvenated for a period of time, and it retains its cell identity so that the organs that we're trying to rejuvenate retain their function and avoid teratomas, cancer.
So is this Goldilocks zone different for different organs, different tissues?
This is kind of the million-dollar question that we're now exploring, that most likely it is. Different cell types will have different potentially even different factors, because we're trying to actually move away from Yamanaka factors to novel, safer factors that will be tissue and organ specific. But even with the Yamanaka factors, yes, we feel that different cell types will have different durations of the safe Goldilocks zone. And we are actually already see indications of this. There was this wonderful preprint out of Alejandro Campos' group in Lausanne, where they showed that if you avoid the liver and the intestine, you can activate the Yamanaka genes in vivo in these transgenic mice for much longer periods. Basically, the problem with activating them for too long is that mice start dying. Like after 3 or 4 days of induction of Yamanaka genes, mice start dying. So they try to investigate, okay, which tissues are actually causing this? And they found out that if you avoid the liver and if you avoid the intestine, you can activate Yamanaka genes for up to a month. And I think 60% of the mice, they activated them for a month and survived. And I think everybody survived up to, I think, day 10 or day 12. My memory is a little vague. But 60% survive a whole month of induction. And this will help us, I think, find different dosing regimens for different tissues that will enable us to have an essentially tissue-specific approach to rejuvenating, tissues that we need to rejuvenate. So I guess the short answer is yes.
Is it proven that stimulating these factors increases the lifespan of these mice?
Right. This was shown in a progeric mouse model that using these factors in vivo, you can prolong lifespan of this progeric mouse model, a fast-aging mouse model, by up to 50%. Basically, they had different control groups. The mice which got the treatment lived up to 24 weeks, the median lifespan, but different control groups lived between 16 and 18 weeks, the median lifespan. So you can depending on which control group you compare to, you have either 50% increase or 33% increase in median lifespan. And of course, this was done in transgenic mouse model in which all tissues had Yamanaka factors. And so you couldn't do a tissue-specific activation. It was either all or nothing. So they established initially that if you activate Yamanaka genes for 3 days, this was mice start dying after 3 days of consecutive induction. So they chose 2 days as kind of the safe activation period. And so the way they induced factors in that model was 2 days of factor induction, followed by 5 days of chase or like washout period, letting kind of the activation of the factors stop and letting the mice recover essentially from this rejuvenation burst. And in that way, they were able to prolong lifespan, as I said, by up to 50%. But now we know that if we avoid the liver and the intestine, we can maybe try a longer period of full body systemic induction of these factors, or alternatively try to approach in a tissue-specific manner where we'll be activating it differently in different tissues and maybe even spread it in time, where you, for example, rejuvenate the brain first without any other organs, and then you follow it with other organs, other tissues, where you induce the factors separately. So to answer your question, I guess, we have this data in the progeric mouse model. We're still waiting to see if we can replicate these results in regular wild-type mice, non-progeric mice. And with this new result from the Ocampo lab, that if we avoid liver and the intestine, we are quite optimistic that some groups soon will be able to produce life extension in a regular wild-type mouse model by using this approach.
Do you have any intuition about why those specific organs are affected in such a different way than the other tissues?
Yeah, well, I think they are already very plastic, and the cell types that are in those tissues, and they're very high turnover of cells in those tissues. So those organs might be very permissive to reprogramming. So after 2 days, you start getting basically loss of cell identity in those organs, possibly. And this is what's causing, teratomas or loss of function. And, without the liver or without the intestine functioning properly, the mice just cannot survive. And so, yeah, I think we should avoid fast dividing tissues in general, I think. Not only because of these risks, I think in terms of rejuvenation, I think those tissues are kind of less of a need to be rejuvenated versus, say, for example, the brain or the muscles or vessels and other kind of critical organs that we think are, at least for humans, are responsible for most of the deaths that we see with age-related diseases.
Well, when you say rejuvenation, you're importing a certain theory of aging.
Am I?
Well,
okay.
I mean, otherwise, how do you define aging? And so I wanted to get your take on what is the underlying theory that informs this approach.
Well, okay. I think the beauty of this approach is that it's not dependent on the theory of aging, because it's very practical and essentially, empirical observation that reprogramming rejuvenates aging hallmarks, cellular aging hallmarks on any cell you start with. Basically, the population that you end up with is rejuvenated on a cellular level. And so we're trying to translate this then on an organismal level for in vivo reprogramming. And it doesn't matter what we think is driving aging or what's happening, because we're just taking this observation, this empirical result, and trying to create a therapy out of it. But of course, I have my own kind of vision of how this is working and why this is working. And to me, it comes back to the epigenetic theory of aging, or the epigenetic driver of aging, which I think is the key driver of the aging process, epigenetic changes that we see with age. And of course, within the epigenetic changes, there are some stochastic changes like random changes, noise, drift, epigenetic drift. But also, more importantly, I think within the changes that we observe with age, epigenetic changes with age that we observe, there is this non-random component, which I think is programmed. Other people think it might be deterministic, but not in a programmed way. It doesn't really matter, because I think at the end of the day, this informs the hypothesis that if you're able to rewind backward these epigenetic changes, which are essentially proceeding in a clockwise manner. That's why we can build epigenetic clocks based on the changes that we see, epigenetic changes that we see. We can have epigenetic clock of people of the same age. And so if you actually rewind them back, you can rejuvenate the organism. You can provide this organism with a more youthful pattern of gene expression. And this is what Yamanaka factors essentially are doing. They're taking the cell back to the embryonic-like state, where in the process, it's upregulating basically the good stuff and downregulating the bad stuff that we see with age is actually moving in bad directions. Basically, with age, the good genes that help us, for example, prevent damage, the expression of these good genes is downregulated with age, and the expression of bad genes like retrotransposons or some other aspects of the cellular machinery that maybe we shouldn't upregulate with age, or we don't want to see upregulated with age, inflammation, for example, actually with age, it is increased. And so basically we're trying to take this pattern of expression back to a more youthful pattern, and this is what Yamanaka factors seem to be doing. And this, I think we are repurposing a program that life has had for, I don't know, millions, billions of years of rejuvenating a new organism. Basically, this program of rejuvenation is reserved for new babies, progeny, which, essentially is your own cell, if you're a female, a mother, egg cell, which has been aging at the same age as the mother, because it forms when the mother is still an embryo. So every baby starts off as a cell of the same age as the mother. But that 30 or 20 or 30, 40-year-old egg after fertilization is actually rejuvenated, like fully rejuvenated. Age is reset. Any damage that it might have accumulated during the 20, 30, 40 years it was present in the mother is cleared. And I think Yamanaka factors are basically, they're responsible, of course, for the stemness and maintaining the stemness of cells, embryonic stem cells. And I think when they're inducing this shift backwards to the pluripotent state, they're also activating this rejuvenating program. And normally this is done only after fertilization, but we're trying to then kind of repurpose this process and use it for our own rejuvenating benefit of an already formed organism in vivo. So I think we have the capacity, our cells have the capacity to significantly rejuvenate, clear the damage, but that capacity is only reserved for progeny, for evolution only wants this done in the context of a new child. But we obviously want it done in the context of already grown organism, and basically we're trying to figure out how do we maybe kind of hack this program, extract the rejuvenating part without the dedifferentiating part, and maybe find out in this process of reprogramming, and this is actually another thing that we're exploring, we're trying to decouple this process of rejuvenation from the process of dedifferentiation, the process of losing cell identity, and trying to get just the rejuvenating part to then apply it in vivo in the organism.
Could you define epigenetics just...
Okay, yeah, probably, yeah, by now, if you don't know what epigenetics is, you might be a little lost following the last 15 minutes of our discussion. But epigenetics is just a generic term of control of gene expression. And there are different mechanisms that control gene expression in an organism. And the problem is we are multicellular. We have very different cell types, tissue types, organs, but all of our cells are, essentially a copy of the initial fertilized egg, and obviously that means they have the same DNA. All of our cells that still have DNA, unlike red blood cells, all our cells that have DNA have the exact same copy of DNA. But obviously they need very different genes to be activated. And actually most of DNA is actually silent, I would say, most genes, compared to the total of how many genes we have in a given cell type, I would say, yeah, most genes are silent. And it is a matter of, epigenetics is a matter of controlling which genes are silent, which genes are upregulated, so that you can actually have multiple tissue types. So epigenetics just kind of is a control program for the genes that makes skin cell a skin cell, brain cell a brain cell. And also its epigenetic mechanisms are responsible for developmental processes. They need to, basically the function of an organ at, say, age 2 years old is different after different organs. But after maturity, also epigenetic mechanisms control how the organ functions, or when it grows, how quickly it grows. And so essentially epigenetics is a kind of control mechanism, both temporal and in other dimensions of how and when genes are expressed or suppressed. And the connection of epigenetics to age is that it's been noticed that during the aging process, the pattern of epigenetic changes is actually very similar. The change in that pattern is very similar across different people. So much so that we can have epigenetic clocks. We can build a clock that just based on the methylation pattern. Methylation is one of the mechanisms that controls gene expression. So based on the methylation pattern found in your skin cell, you can tell how old a person is. So without knowing anything at all, you can tell, okay, this skin cell comes from a 40-year-old or a 60-year-old or a 20-year-old. And this pattern is universal, for all humans. Moreover, it's actually now established that there is a universal pan-mammalian methylation clock. So it's actually evolutionarily conserved, at least in mammals. And probably, different species might have different epigenetic aging regulation, but our field has been, with obviously good reason, more concerned with mammals, because ultimately this informs what we can do for humans. And they found out that there's conserved elements of epigenetic clock across different species, scaled for, of course, the speed or the life history duration. A mouse essentially goes through its life history 40 times or 30 times quicker than a human does. And so its methylation age or its epigenetic clock is basically moving in a very similar direction or a very similar manner as a human clock, just scaled for that speed difference. So where was I? Sorry.
On that note, this research is mostly being done in mice when it's in vivo. And what are the major hurdles you see to this being translated to humans?
Well, we're still, I mean, if we're able to rejuvenate mice, then I don't see major hurdles of translating that to humans, because this process, as I said, is a very evolutionarily conserved process. The epigenetic changes with age, reprogramming, you can use human or Yamanaka genes in a mouse and they will do their thing perfectly, rejuvenate mouse cells and bring them back to pluripotency. So the biggest hurdle right now is how do we actually first do this in a mouse? We have as I mentioned, the result in progenic mice, fast-aging mice. Now we have to do essentially replicate or find out how can we do this in normal wild-type mice. And then once we figure that out, a matter of scaling that to humans, I think, is just a engineering problem, a genetic engineering problem. So I don't see major hurdles. I just see kind of small hurdles of, once we figure it out in mice, small hurdles of translating that result to humans.
What's the time scale you see for that translation?
Right. Well, I think we're pretty close to figuring out some aspects of reprogramming, like tissue-specific reprogramming, that could help with specific pathologies, like problems, for example, Alzheimer's, right? And so translating those aspects into therapies, I think, will be accomplished relatively quickly. Within 3 to 5 years, I think we might see already some clinical trials of applying the partial reprogramming approaches to some diseases or some pathologies, for example, or trauma. And people are looking at various things from macular degeneration, eye problems, age-related eye problems, to nerve damage. And we're in Boston. Obviously, David Sinclair's lab has been working on this paradigm, partial reprogramming paradigm, for several years. And I think they mentioned that they hope to get into the clinic for either nerve regeneration for people who suffered some spine damage or nerve damage, or for eye diseases, basically within several years. And I'm not sure about their progress, but hopefully, soon enough they will be able to enter the clinic. With our therapies, we hope, if everything is successful in the mouse models, I hope that within 2 to 3 years, actually, we might be able to get to the clinic for one particular indication that we're looking at. And for Alzheimer's, again, if everything goes well, I think within 5 to 6 years, we might also be able to start start clinical trials for Alzheimer's. In terms of a more systemic rejuvenation therapy for humans, I think this will need to be built on a tissue by tissue level. And once we figure out basically there are enough tissues that if we rejuvenate, or if we slow down aging in those tissues, then we can see a systemic effect of either a slowing down of aging or even rejuvenation of an organism. And translating those findings, I think, we could see within 10 to 15 years. And also, I think there's going to be a lot of overlap between, curing diseases or trying to use these paradigms in models of disease like Alzheimer's, and then trying to use the same paradigms as preventative therapies to prevent the same diseases, or in general age-related diseases. And you can have clinical trials where one of the endpoints could be the delaying of any age-related condition or major age-related conditions. Like, I think the TAME trial, the metformin trial, is using those endpoints as basically trying to tease out, can metformin slow down the aging process? And I think the same can be applied to partial reprogramming therapies once, of course, we have animal data that will allow us to think that we can rejuvenate animals, we can slow down aging in animals, and then we'll start translating that result into humans. But to summarize, I think relatively quickly, like 5 to 10 years, I think we'll start to see exciting therapies being tried in the clinic. pending positive animal data, you basically, scientific risks are always there. You get very excited about some results, but then, science sometimes kind of teaches you lessons when you shouldn't be too excited. So, but what we observe, judging by what we observe now, I'm pretty optimistic, and hopefully, the optimism will continue, and science will support that optimism with results soon enough. And there are many different encouraging results coming from different labs, looking at different aspects of reprogramming. And I think synthesizing all that into therapies is also happening. Obviously we have all these new companies forming with a lot of investments like Altos Labs, Retro, New Limit, and a lot of investments, hundreds of millions, billions of dollars going into this area precisely because there's a lot of optimism by leading scientists in the aging field that this is a very promising approach. I don't think the aging field had an approach as promising as partial reprogramming because of, to me, the epigenetic nature of aging and the fact that reprogramming is essentially operating on the epigenetic level. So,
what would you have to see to try it on yourself, to take these, whatever the final result is, and what would convince you to say, okay, I'm in, I'm doing this?
Well, I think like with any therapy, I would have to weigh the risks and the benefits, and I would have to see enough benefits first in animal models and probably in some human clinical trials as well, that there are benefits in terms of rejuvenation, in terms of slowing down of the aging process, and that the risks are minimal, that the therapies are safe. And just like with any drug development, we'll have to first prove it or establish to a reasonable degree that the therapy is reasonably safe and the benefits are there for you to justify it. So as soon as, I think I would be willing to be one of the first patients to try this therapy for my age group once we established its safety and efficacy in disease models first for, I guess, Alzheimer's patients or some rare disease patients. But once that's established, happy to try.
Do you think that there could be an issue with the weak link in a chain? So assuming that if we just use that mouse example, that you're able to rejuvenate all the tissues except the liver and the intestines, do you think that even if you have all these well-functioning organs, the organism itself as a whole will still die at a normal lifespan because of critical failures in one of those two organs or the combined function thereof?
Well, I don't think the liver or the intestine are limiting our current aging, or at least in mice, I guess, because we're looking at the mouse model. Humans, for us, it's heart disease is the biggest killer, cancer, and then Alzheimer's and dementias. And I think partial reprogramming can make beneficial therapeutic effects in all of those areas if we are able to, if we rejuvenate tissues in which we see, for example, cancer, we upregulate mechanisms that are actually able to prevent cancer from occurring in the first place on the cellular level, prevent, mechanisms fixing DNA breaks. And this is actually something we observe in cell cultures, that cells that undergo reprogramming, even partial reprogramming, the markers of DNA damage, like markers of double-strand DNA breaks, are significantly reduced. So potentially by partial reprogramming, we can reduce the chances of cancer happening in tissue-specific manner. And also that will hopefully translate in a systemic effect. And systemic effect, I mean reducing our biological age, which is a measure of essentially our mortality risk with age. If we're able to reduce our mortality risk that with this therapy, we would expect a person who has been treated to significantly extend their lifespan. So if we're able to see this in animal models or later in the clinic, I think this is a strong enough signal that this therapy can be expected to significantly extend health span and lifespan. And in terms of what else might fall out if we're able to kind of rejuvenate organisms and make them live longer by avoiding cancer and dementias, what else could they start dying of, 150 years in the future? I'm willing to find out, and not really worried about that, or at least not worried enough not to try this kind of therapy.
Well, I hope we all find out very soon.
So do I.
Thank you.
My pleasure.
Thank you for joining us today. We hope you join us next time as we explore and elucidate the frontiers of science and technology with the thinkers creating the future.